So welcome to this edition of the ERGA's BioGenome Analysis and Application Seminars. So it is my great pleasure to welcome Orly Razgour. She's a senior lecturer in ecology and, at the University of Exeter, and she's leading the Global Change Genetics group. And thank you, Orly, for accepting giving this talk. And I'll also introduce Chris Barratt, who's a postdoctoral researcher in iDiv, the German Center for Integrative Biodiversity Research, and also a guest researcher at the Naturalis Biodiversity Center in Leiden. And today the topic is devoted to biodiversity applications, and it will focus on how can we use genomics to inform biodiversity conservation, to predict adaptation and inform biodiversity conservation under global change, which is a very important societal question right now. So Orly will present this topic during 20 minutes, and then Chris will present a toolbox that they developed called Life on the Edge. And then after this 40 minutes, we will have 20 minutes for a Q&A, and I'm very much looking forward. Thank you again, Orly and Chris, for agreeing to give this seminar, and I'm looking forward to hear your talks.
Thank you, Jose, for inviting us and to give this seminar. And good morning, everyone. I'm just going to share my screen. Okay, so good morning, everyone. Today I'm going to talk about how we can use genomics to inform biodiversity conservation under global change. And I'll present some of the work that's been carried out in my research group, the Global Change Genetics Group. Okay, so we're all aware of the impact of global environmental changes that have occurred over the past few centuries in terms of anthropogenically driven climate change and extensive land use changes, resulting in habitat loss, fragmentation, degradation. Now, all these kind of environmental changes have had major impact on biodiversity and pose a really major threat to global biodiversity. But it's not just wildlife that's affected by it, it's also the ecosystem services they provide. And as a result of that, it's really affecting our survival and our global food security. These kind of global environmental changes are posing new pressures on species that are forcing them either to move away from where they're currently found or to adapt or acclimatize to these changes. The newly emerging genomic approaches over the past decade and a bit provided us with new tools to be able to better understand these changes and be able to better inform conservation management. So genomic approaches can be used to monitor biodiversity responses to these global environmental changes. And as part of the GeoBond genetic composition group, we recently published a set of essential biodiversity variables that look at how genetic composition have changed over time. So this is to enable us to monitor responses to environmental changes that have occurred by looking at genetic diversity, genetic differentiation, inbreeding, and effective population sizes. However, the problem here is that we actually don't have these long-term time series data a lot of the time to be able to see and monitor these changes that have occurred over time. So this is something that we're slowly starting to collect now. So instead, commonly, biodiversity vulnerability to these kind of global environmental changes is assessed through using correlative climatic niche models, also known as ecological niche models or species distribution models. And these kind of models, what they do, they use information about where the species are currently found, the environmental conditions where they're found, and then use modeling approaches to project where suitable conditions occur either across the species range or under changing climatic conditions. This kind of approach are really widely used. However, they've been criticized also for being oversimplistic and for disregarding important mechanisms that affect species vulnerability to these environmental changes. And for example, these can include evolution and adaptive variation, physiology and thermal tolerance, population dynamic, dispersal ability, and species interactions. So research carried out in my group tries to address some of these gaps in our ability to properly predict vulnerability to environmental changes. And we use it through integrating different approaches. So we combined together genomic tools with ecological research and different modeling approaches to try and understand the ecological and evolutionary responses of biodiversity to these global environmental changes. And today I'm going to show you a few examples of the kind of approaches that we've been developing. And then Chris will go into more details about how he's taken that and turned it into a bioinformatics toolbox. Okay, so a lot of the work I'll be presenting focuses on bats, and this is because bats are widely distributed. They're very diverse in terms of the number of species, as well as the ecological roles that they play. But bats globally are under threat, with 25% of species categorized as under the IUCN Red List under one of their threat categories. And in fact, many other populations of bats have also experienced decline. But beyond that, bats are also a host of zoonotic diseases, including viruses. So it's quite important for us to be able to understand how these global environmental changes affecting the distribution of bats, and as a result of that, the risk of spread of zoonotic diseases. So we recently carried out a systematic review to assess bat responses to climate change and see where that literature currently stands. And what we found is that many papers have been published on spatial responses in terms of range losses, range gains. There's also been quite a lot of papers published on demographic responses in terms of changes in population sizes. But there's a lot less literature on genetic and physiological responses. So these are currently a missing gap in our understanding. So I'm going to start by talking about how we can incorporate adaptive variation into our vulnerability assessment. And the idea here is to use genomic data to inform our ecological niche models or species distribution models, but also use the genomic data to identify adaptive variation to climatic conditions and use this information also to predict the potential for evolutionary rescue. Now, when I talk about evolutionary rescue, what I talk about here is populations that are maladapted to future changes. They'll be able to survive as a result of gene flow coming from locations there, or populations that are adapted to warmer and drier conditions, i.e. the conditions that would occur under future climate change. Okay, so I'm going to show you how we can use this framework with two case studies. So this is two bat species, Myotis escalari, Myotis crypticus, that are found around the Mediterranean basin. One of them is restricted to the Iberian Peninsula, that's Spain and Portugal, and the other one is found at the northern parts of the Iberian Peninsula, southern France and Italy. And we generated genomic data sets for these bats, around 20,000 SNPs. So first we would need to identify climate-adapted individuals. And for that, we use genotype environmental association analysis to identify genomic regions that are potentially associated with high summer temperatures and summer rainfall. And these are two variables that are important for bat survival and reproductive success. So once we identify these genomic regions, so in this case we identify 32 and 38 SNPs, we then plot our individuals across the ordination space to see where they fall relative to these two climatic variables of interest, based on their genetic makeup in those potentially climate-adaptive SNPs. And then we divide our individuals into those that are associated with hot and dry conditions in white versus those that are associated with cold and wet conditions in grey, based on their genetic makeup. And we also have individual genotypes that are more intermediate that we don't include in this analysis. Okay, then we directly incorporate that into our models. So the models you see here on your left were generated using all the location records for the species. In this case, it's Myotis escalari that is restricted to the Iberian Peninsula. Blue colors represent low probability of occurrence. Yellow or orangey colors, high probability of occurrence of the species. At the top under present conditions, and at the bottom under projected future conditions for 2070. And if we compare these two maps, we can see extensive projected range losses in the Iberian Peninsula where the species is currently found. But then what we did, we created different models. So we created separate models for individuals that were identified as adapted to hot and dry conditions, and these are in red, and separate models for individuals that are adapted to cold and wet conditions in blue. And if we compare the two maps at the bottom here, which are under projections under future conditions, we can see that when we incorporate adaptive variation into our model, we have reduced range loss projections. Now, this is quite important because that means that a lot of the projections that we're currently using to inform conservation may be over-predicting range losses. And this can result in misguided conservation efforts and priorities. Okay, then we use this genomic information to assess the effect of landscape barriers on evolutionary rescue potential. So how we do it, we first use the landscape genetics approach to relate genetic variation into different landscape variables that can affect movement. So what we want to identify is the landscape variables that form barriers to gene flow and connectivity between populations. And these two bats are forest bat species, so it's not surprising that the main landscape variables affecting gene flow are forest cover and topography. So then we use this information to project the potential movement density between our populations that we know are adapted to hot and dry conditions. So they're adapted to conditions similar to what will be under future climate change, into those populations that are maladapted because they only have individuals that are adapted to cold and wet conditions. That it's here in grey. So we project the potential gene flow from adapted to maladapted populations based on the effect of the landscape on genetic connectivity. And what we see that there is a potential for evolutionary rescue. However, landscape barriers to movement are likely to affect evolutionary rescue. Okay, so now I'm going to move into a different topic and looking at other type of conservation application of genomic approaches. So in this case, is using genomic approaches to identify historic population declines. So wildlife populations have been declining dramatically in the past few decades. The Living Planet Index has identified around 70% declines in the relative abundance of wildlife populations between the 1970s to today. So these are really substantial population declines. However, the real challenge is to understand the magnitude of these declines relative to historic population sizes. So what did the population actually look like pre-industrial level times? So before all the extensive land use changes that have occurred in the past few centuries. The other challenge is also to understand what are the actual consequences of these declines. I'm going to show you now how genomics data can be used to help inform our understanding. And the reason why we can use genomic data within this context is because the genome of an individual contains information about ancestral genomes and the evolutionary forces that shape them. And as a result of that, we can use information from current individuals to understand the genomic makeup of past populations and estimate the sizes of past populations. So here our aim was to identify historic population changes and their drivers using genomic data. And what we're going to focus on here is the Western barbastelle bat, Barbastellus barbastellus. So we focus on this bat because this is a bat of European conservation priority, because it's identified as threatened across its range and has actually experienced population declines and has been lost from some areas. Now, the species is closely associated with old growth forest habitat, so habitat that's been declining across Europe. Now, there are anecdotal evidence that suggests that bat populations have declined dramatically in the past few centuries in response to land use changes and persecution. However, there's a lot less empirical evidence to support this decline. And this is really where genomic data can help fill the gap. So we generated a genomic data set for these bats with around 40,000 SNPs. And our main focus was on the British bat population of this species. So first we looked at landscape drivers of genetic diversity. And what we found is that levels of inbreeding decline with increasing forest cover, in particular broadleaf forest cover, and increasing habitat diversity. So what we see here that when you have areas that have over 20% forest cover, you have higher genetic diversity and lower levels of inbreeding. We then looked at landscape drivers of connectivity using the landscape genetics approach. And what we found that genetic connectivity between British populations of British colonies, of the barbastelle bat is reduced as artificial lights at night increase, but the genetic connectivity is increased with increased coverage of broadleaf woodlands and riparian vegetation and rivers. So what we see here that these land use changes that have occurred in the past century has had an impact both on their genetic diversity and genetic connectivity of this species. So then we wanted to look at evidence of historic population decline. So for that, we used approximate Bayesian computation, model-based inference of demographic history. And we compared different scenarios of what may have happened. And we compare all these scenarios to a null model whereby there's been no change in population size. So we look at scenarios whereby the population have increased over time or decreased over time, and when did these changes have occurred, whether they are recent or more historic changes. Now, the model that received the highest support was a scenario whereby both the North and South British populations of these bats have declined dramatically, so by 99%. And these declines were estimated to have occurred at around 300 to 500 years ago. So what happened around this time period? Well, we're looking at the mid-15th to late 17th century. And this period in Europe is known as the Age of Discovery. This is where the first colonial expeditions have happened. So it's really the birth of colonialization. This kind of colonial explorations required extensive shipbuilding. In order to build these ships, they needed large quantities of outside mature oak trees. So around this period, there's historic evidence of decline in the coverage of broadleaf woodland, and in particular decline in those mature older oak trees. Now, these trees are also the trees that are favored by maternity colonies of the barbastelle bats for roosting. So what we can see here is a link between environmental changes that have occurred as a result of these colonial explorations and the decline of these bats. So colonialism, also bad for bats. Okay, I'm going to finish very quickly by just introducing how we combine this kind of information together to guide conservation management. So this is an integrated framework that we developed to identify which populations are under threat from climate change, to be able to understand which population requires conservation effort and what kind of conservation effort they will require. Now, we focus here on three measures of threat or vulnerability to future changes. So the first is exposure. So how will conditions change in the future? The second is sensitivity. So can they cope with future changes? And this is based on their genomic makeup, both adaptive variation and neutral variation. And the third aspect is range of potential. So can they move away? And combine this information together in the risk assessment framework to divide populations into those that are under lowest to highest risk or vulnerability to future changes. And then we can use this information to also divide management and conservation interventions. And these range from least intensive to more intensive. So let's start by quickly looking at just what's happening here at the lower end of vulnerability. So we have here populations that are found in areas where conditions are not going to change as much. They're still going to stay relatively suitable for them. They're less sensitive to these changes because they already have adaptive variation that is associated with hot and dry conditions. And in terms of the movement ability, they will retain landscape connectivity, so can move if they need to. So in terms of the conservation management, what we may want to do is create protected areas, improve the habitat in these areas to make sure that we really bolster these populations. So we have these large source populations that can form a source for colonization of other areas and the spread of adaptive variation to other maladapted populations. Now, at the other extreme end, at the high vulnerability end, we have these populations found in areas that will become unsuitable under future conditions. They don't have the adaptive capacity that will enable them perhaps to respond to these changes. So they are maladapted to these changes, so highly sensitive, and they're found in areas that their landscape connectivity will be reduced and they won't be able to move and shift their ranges. So in this case, we need more extreme conservation efforts like either translocation into more suitable areas or taking them into captive breeding. But these kind of extreme measures of conservation, or very intensive measures, are not really feasible when we think about the extent of biodiversity that will require management under climate change. So perhaps a better focus may be more at the mid-vulnerability area where we have populations that are found in areas that will become perhaps unsuitable, but they may have adaptive variation that's already associated with warmer and drier conditions. And what we may want to do in terms of conservation effort is increase landscape connectivity in order to enable them to either move away, but also enable this movement of individuals that will enable gene flow of adaptive variation into these adapted populations. Okay, so Chris is going to talk next about how he's taken these kind of approaches, put them all together into a toolbox live on the edge. And he's also using these approaches to actually applying them to around 100 species of small mammals, reptiles, and amphibians across the Palearctic and Africa. So this is just to thank the different collaborators and funders of this work. And I'm going to stop sharing and give the floor to Chris.
Thank you, Orly. So, Chris, the floor is yours.
All right, thanks. I'll just also share my screen. Okay. You can see everything, right? And you can hear me? Good. All right. So hi everybody. Thanks, Orly, for giving a really nice introduction to the topic. Usually when I give these kinds of talks, I have to do a lot of the intro stuff as well as the actual nuts and bolts of the toolbox. So this is quite cool. This is quite unique. And yeah, thanks to the organizers as well for inviting me, because this is really nice. So I'm going to talk about Life on the Edge, which is a new toolbox to predict population vulnerability to global change. And I'm going to give you a kind of overview of the project in its entirety, but we're going to delve into the specifics of the actual toolbox. So it's in big collaboration with Orly, among other people as well, based in the US. So Brenner and Malin in the US, and then in Europe, Renske, Sebastian, and Hjalmar. And the project's supported by the DFG in Germany and iDiv, and more recently Naturalis in the Netherlands, which is where I'm talking to you from today. So the motivation for the project is really that the archives are really full. We've got lots of genome-wide data that's being stored. It's accumulating rapidly in repositories for many different types of species, and they're getting stored in things like the Sequence Read Archive and the European Nucleotide Archive and the Data Bank of Japan. These are all typically kind of population genetic or phylogeographic studies, and they can range from kind of RAD-seq or DD-RAD style data to whole genome sequences. The key is that they're typically used once. So we tend to make a nice study. It's someone's PhD or postdoc or a project for surrounding that, and it will get archived and often never looked at again in any great detail. So the motivation for this project was to make use of these kind of vast archives of information. As well as all of the data that's available and accumulating, there's lots of genomics informed methods to prioritize vulnerable populations. But these have typically been quite species specific. So, I mean, I'll highlight a couple of Orly's publications here in the center of these four. There are many of these methods, but they're a bit fiddly to fit to other species. So that was another main motivation for this project. So the main idea was to build a generalized framework to predict population vulnerability to global change. And the key is to make it applicable to any species and any geographic area. So we're not kind of locked into a specific system. And the second motivation was also just to make this modular so that we can easily edit and expand it in future. So what we've made is kind of the backbone for something which we hope is going to grow into something even larger and more comprehensive. So this is just an updated figure of the overview of the framework. The main aim on the left is to identify populations under threat from global change. And as Orly nicely presented before, we've got kind of these three main aspects. We've got exposure, we've got sensitivity, and we've got what was called range shift potential, but has now had to be renamed because of a reviewer that didn't like the title of that that aspect. So we call it movement barriers now, but it's essentially the same as mentioned in Orly's publication. We use distribution models and environmental dissimilarity between current and future climate projections. We look at adaptive regions of the genome. We look at things like genomic offset as well. And we look at standing neutral genetic diversity too. And as well as this, we look at movement barriers. As Orly was talking before, we look at the potential connectivity between populations as a way to escape changing climates if you can't adapt to them in situ. And we put all of this together to make a prediction for every sample or every population that we have across the region. So you can see on the right here, you will have a kind of a population vulnerability kind of map of all of the populations, which is colored from low to high. And this is flexible for any data set that you've got. So the idea is that you have the input data, which will be population or individual level genomic data. Typically, it's going to be kind of Plink formatted. So you've already processed the genomic data and you put it into Life on the Edge, along with the georeference coordinates of all of the samples and the populations. And you need to supply some sort of environmental predictor data for your distribution models and also for your genotype environment association analysis. So this needs to be ecologically meaningful. So you need to think about what this means for your specific species. So you shouldn't just blindly put everything that you can think of in. So then you put it all into Life on the Edge, which is this magical machine. And what it will do is there's a parameters text file which has about currently 60 parameters across different analyses, and each species will have a line with 60 parameters. And Life on the Edge will use all of these wrapped in the internal functions of the software in R, Bash, and Julia to give you kind of standardized outputs across species or for the given species that you analyze, but these will be comparable across different species that you do. So the nice thing about it is that it's super structured, it's super standardized. There's a nice summary and there's a nice summary and a log file, which is spitting out everything that you need to know, all of the information, so that if anything funny has happened or you get some unexpected result, you can track everything back. So it's definitely not a black box. And you get this nice summary, which is comparable across the different data sets that you put in. So it's containerized in a singularity container, which is kind of the main machine for running the whole thing. So it will basically run the main aspects of Life on the Edge using a number of different functions. And I won't go through all of these in detail, but basically you've got quite some functions to prepare the data before you go ahead. Then you've got some functions that focus on the spatial data itself, and then you've got some that focus on the genomic data, and they match different parts of the framework. So exposure, sensitivity, and movement barriers. So there's a lot of stuff in there, and it's basically all wrapped up in a wrapper script. So you can run the whole pipeline or the whole toolbox on a species with a single line of code if everything's stored in the right places. And you can loop this across different species or different data sets to get your outputs. So as I said, I won't go too into detail with these functions themselves, because we'll go a little bit more in detail in the next slides. But essentially, you get an output directory for each species, and it will be named with the name of the species, and you will get a folder for exposure, a folder for sensitivity, a folder for movement barriers. And within each of these, there are subdirectories with the relevant analyses, and you'll get a final population vulnerability folder as well. And as I mentioned before, your summary and your output logs as well. Just another note on the container, and the reason for that is that it's got a lot of dependencies. There's a lot of different analyses in here, and if it's not containerized, there might be some trouble with different versions of different packages and different softwares. So it's all clumped together in a nice singularity container where you basically run a pre-setup and install the relevant packages into the container, and then it's good to go. And it's typically for HPC kind of cluster use, obviously. So you can run it in its entirety from the beginning to the end, or you can run specific parts. So the user's in control of everything. You don't just, well, you can just run it, but yeah, you should be in control of the parameters that you need to set. If there are parameters that are missing that are important, it will fail. So you need to think about that, about what you're going to parameterize. But you can skip sort of like different steps. You can skip auto prep of the spatial and environmental data, for example, if you already have very well curated data yourself, or you've already got nice distribution models, you can plug those in to the framework. And it can run multi-species, as I've mentioned, so one line per species. So it's all run in parallel via array jobs on a cluster. But you could do it on time series data, for example, if you've got repeated sampling of populations over time, or you could do it for different projections of climate change, different global circulation models, different years of projection, different shared socioeconomic pathways. So there's a lot that you can do, and that's why it's been parallelized to kind of inspire that. Basically, there's a step at the beginning to beat data into shape. So at the beginning, you will supply the predictors and the georeference data, so to make the environmental data ready. And Life on the Edge will extract and prepare everything for you. There is an option to auto download environmental data yourself from WorldClim, for example. But you can do this. You can do this, or you can provide the predictors that are of relevance to the species that you're focusing on. So you will have a bunch of environmental predictors, and I'm showing the example that I show here is all an East African frog, which I know very well, and it's very close to my heart, so I'm very familiar with these data. What it will also do is kind of do automated species occurrence data downloads from the GBIF repository using the R package associated with that. There's some cleaning steps and some spatial thinning to account for spatial autocorrelation, or you can provide your own clean and trusted species presence data if you've got a comma separated file with points, for example. And the same goes for background data as well. So pseudo absence data for your distribution models. You can get it to auto generate the background data, like on the right at the bottom here, or you can provide your own. The key part is that all data prep steps for the spatial data here are parameterizable for spatial resolution, extent, for defining the predictors, different projections of years and sort of different scenarios of what's going to happen based on climate models, and also for spatial autocorrelation terms as well. So we're moving into the framework itself now. We've beaten data into shape and it's all ready to go. Typically, you'll build some distribution models, and we use Biomod 2 for this. Again, it's highly parameterizable with the kind of algorithms that you'll use for distribution modeling and how you evaluate them to retain good models. So what are your criteria, whether they're strict or relaxed? And we typically use the area under the curve and true skill statistic to evaluate these. And you set a threshold which you would like to evaluate a good model. And with these good models, you'll make an ensemble species distribution model, like shown on the left here, and you'll project this in space and time with your good models to look at a future prediction. So you'll get a sense of where the species distribution will lie in the future. And with this, you can then calculate environmental dissimilarity for the species distribution model. So you can see in orange regions where the range of the species is gonna reduce a little bit, and you can see areas of expansion in green, and white areas are kind of no change at all. So we just use simple raster overlay for that, but we're also able to look at specific environmental variables across our populations to kind of extract the changes in each point as well. And we use that to quantify the exposure per population. So we've also got a couple of methods for looking at local adaptation. So we've got a univariate method and a multivariate method. So again, it's parameterizable. You can decide how strict or how relaxed you are with what you're going to call a candidate SNP. But essentially, you'll get a bunch of SNPs like on this plot here, and you'll get they'll be basically listed by the framework. So once you've got this list of potential candidate SNPs, you can use these, like Orly showed in her PNAS paper, to categorize individuals. So this has all been automated, so that you can basically parse individuals into kind of different categories, based on ecologically what they mean for your species. So it can be hot, dry, and cold, wet, or intermediate, like Orly mentioned. But then we can map them in space, in geographic space, and we can get a sense of which populations are more hot, dry adapted, which ones are cold, wet adapted, and which ones are a bit more intermediate. And we can also look at genomic offset as well. So that's a recent introduction, based on a reviewer comment. So we've also got ways to look at potential maladaptation to future kind of environmental conditions. As well as the adaptive sensitivity, we can mask out the putative adaptive SNPs, and we can look at just the neutral data set, and we can then do some simple stats to calculate heterozygosity in each population, and use that as a measure to look at neutral sensitivity as well. And moving on to the penultimate part of the framework, we're looking at population connectivity. So it's now called movement barriers. It was range shift potential in all these papers. Basically, there's a way to use electrical circuit theory to map potential gene flow between your populations. And what you'll do is you'll select some ecologically meaningful variables, and you'll give them a given weight. And you can sort of decide this based on your distribution models, for example, of variable importance to the species. You make a cumulative resistance surface, ranging from high to low, and then you'll plug in your actual data and you'll model the connectivity between populations. And you can see the plot here on the right shows that it's a little bit higher on the kind of northern populations, and in the southern populations here, which are a bit more montane, it's quite disconnected. And lastly, we're making all of these all the way through this. We're making tables. I mean, it sounds like it's really fancy, but actually all we're doing is making tables for everything, and we're recording everything per population. So each row is a longitude, latitude, and we're for exposure and the two sensitivity metrics and the movement barrier metrics. We're basically just having all this data together and using that to make a population vulnerability metric. And typically I use the mean of the average, sort of the average of the other metrics, but you can also weight that and combine it in different ways if you want to. And then we can plot it in space. So you can plot each individual metric, or you can also plot the vulnerability of each different population. So you get a sense of which populations might need more help. If you've got limited resources and you need to select some for prioritization, you might use something like this to do so. As I said, it makes a log file and a PDF file all the way through. So it's like a summary of everything. So you can keep track of everything that's happening. I'm using this, just to wrap up, I'm using this for some species specific conservation questions. So we've got a project in Germany looking at selecting suitable donor and recipient populations for translocations of the midwife toad. The idea is that if we need to move populations around to help kind of bolster genetic diversity, we don't want to grab a bunch of maladaptive individuals and put them in the wrong place. So we've got the active project with the NABU in Germany, but we've also I've got several collaborations with people working on a variety of taxa, looking at identifying hotspots of vulnerability to prioritize conservation. Secondly, I'm looking at multi-species. So I've got a lot of data sets that I'm looking at to kind of look at congruence across many different species in Europe and Africa, and look how that links with the protected area network. And the future developments for this work is starting to bring in functional genomics. So I wrote an ERC starting grant proposal recently to look at bringing in genetic load into this framework as well. There's loads of super cool ideas for biomonitoring with time series data. So this could be with eDNA or whole genomes, for example. And as mentioned before by Orly, if we can start to link demographic declines with population vulnerability, we could also start to bring in the historical perspective of populations into this as well. So the code and the preprint are online. It's in the second round of revision. And so there's going to be some minor changes based on the revisions. So the website's going to be updated a bit soon, but it's mainly just function names and things that are going to change, and a few new funky additions. So yeah, again, I'd like to thank you for the invitation. It's super cool to present and to be given the opportunity. And if there are questions, I guess for me or Orly, then I'll stop staring at my screen. Thanks again.
Excellent. Thank you both for your wonderful talks. I remind people to post their questions in YouTube. And so we have here one first question from Flavia Nunes. I'm wondering if life on the edge could be easily applicable to marine species. What challenges might we expect to have in getting realistic ocean parameters for different processes in pipeline?
Mm-hmm. Yeah, I mean, that's a good question. Yeah, indeed, I showed terrestrial species. I do have someone that's potentially using it for corals. I'm not a marine person. I don't know much about the ocean. But there is, I mean, everything is flexible. And the principles of all of the analyses can be applied to environmental predictor data in the oceans as well. So you can also model gene flow as well quite easily and look at GEAs. So, I mean, if you want to write to me, I can put you in touch with the person that's also working on this too. And feel free to write me for specific things. I mean, I'd be super interested to get it applied to different systems as well. So.
Thank you. So we have another question from Teresa Manosaki. Amazing talks. Are there groups of species with higher or lower chance to predict vulnerability based on the prior knowledge? And can we have an estimate of the confidence of the output?
I don't want to answer all the questions.
Orly.
I want Orly to also jump in.
I mean, I can start just by mentioning something about species that perhaps we may want to focus on predicting vulnerability in that way for. So a lot of the idea behind this work was looking at those longer living organisms with slower generation time. So these are species whereby we don't expect necessarily that new mutations will arise in the population, and as a result of that, they'll be able to adapt to climate change through novel adaptations. But we more expect that the generation time is just simply too slow to really have these kind of responses. And instead, what they will require is gene flow coming from other populations to adapt. And hence the focus here is a lot of it is about movement potential as well. And these are the organisms that are likely to also be more sensitive and less able to just adapt in situ if they don't already have adaptive variation that's associated with the conditions that are changing.
Thank you. We have here a couple of questions from Julia Verba. Great talks for Chris. Is this tool applicable for species with poor data available? Is there a way of dealing with that? For example, not many occurrence points?
I mean, yeah, I think there are. I think you have to be a bit careful. I think you need to be a bit careful because it'll affect your distribution models and it'll also affect your GEA analysis too. One of the main things I mean, Orly and myself have discussed this in depth, is that you really need sort of like at least good coverage of the range of a species to kind of capture the variation within the species. So if you've got a range that's, I don't know, like the whole of Europe, and you've only got samples from Portugal, then it might be tricky to get the full sort of like scope and the full sort of signals of local adaptation. Yeah, so
I guess it would be important to have a good coverage of the environmental variation as well. That is
yeah,
presumably important for the
yeah,
species, right?
yeah. I think it's quite fundamental as well. I think
And
it's
related to that, I have a question about the types of data that you consider more appropriate for this. So, of course, when people are doing population genomic studies, there's a bunch of different types of data that can be analyzed, different sampling schemes. So you can analyze at the population level. So lots of individuals from different places. You can have single individuals from different places. You can have individual genotyping data. You can have eventually pool-seq data from populations where you can derive allele frequencies.
Yep.
What would be sort of the ideal sampling and data collection scheme that you would deem appropriate to increase the potential of finding this properly these correlations and applying this method?
I mean, the main thing is making sure that you've got sufficient signal to detect local adaptation if it's there as well. So that necessitates, I mean, if you've got whole genomes, that's the best. And if you've got multiple individuals per population, we often don't have that, do we, Orly? We're often dealing with, species that are tricky to sample, tricky to, So I think, yeah, you have to be careful with the data that you're putting in. So the stuff I'm working with is mainly sort of dd-rad-seq data and a handful of whole genome sequence data. I'm not even delving into pool-seq data because of the problems of estimating genetic diversity with pooled individuals. That could be something to develop, but maybe Orly wanted to add something.
Yeah, just add to that the other thing to consider is also the sampling scheme. And it's quite important to actually sample across the environmental gradient where the species is found. Because if you're interested in adaptation to environment, or climatic gradient, for example, in terms of impacts of climate change, what you really need to sample is across the climatic gradient. Otherwise, you're simply not really capturing the adaptive variation.
So we have here another question from Julia for Orly. Do you have examples of your results being applied in practice in conservation?
Yeah, so I have, so for example, the work that I showed about using genomic approaches to identify historic population declines, I gave an example of this bat species that we've been working on. And currently the nature conservation authorities in England are using this information to define the favorable conservation status of the species. So this is to understand what the aims of conservation are. And this is what's going to guide conservation policy and prioritization of conservation efforts for this species. So they're using the information we've given on historic population sizes to know where they would like to see the populations in the future, as well as the information we gave on population decline to prioritize the species given the fact that they've been declining so dramatically. So there is a lot of interest to use this kind of data or genomic informed data in conservation. The challenge is being able to translate it in a way that policymakers and conservation practitioners will understand, because a lot of them don't necessarily have a background in population genetics. Their background will be more in perhaps ecology, in the good case, sometimes even not in that field. So it's about being able to translate the results in a way that they can actually use them. And I work a lot with conservation organizations to help translate these findings and to help with creating these links with policymakers.
So I don't think there are more questions in YouTube, but I have a couple more questions. See you. So, Chris, you mentioned that you can incorporate time series data. Does this mean, for example, I'm seeing, for example, the potential of using museum samples, for example, from the past to inform your models. Do you see this as something that could be feasible? And does this imply having also time series of the environmental data so that your correlations are put in the perspective of the potential temporal changes in environmental conditions over the last century or something like that?
Yeah. So I mean, I haven't done it myself yet, but the framework lends itself to that. Um, I mean, I guess with sort of museum samples and sort of ancient DNA techniques, you need to also be careful of kind of DNA fragmentation and the effect that might have on your estimates. As far as the environmental data goes as well, I mean, that's also pretty tricky to come by as well. But if you had a super local scale sort of study, you could probably do something quite cool with that. I don't know, like things like the North American breeding bird data, for example, which is that would sort of lend itself to that quite well. Yeah, so I mean, I haven't done any time series data analysis with it, but what you basically you would treat it as you would run each analysis as a different kind of species, like I showed, in the params, and you can get an estimate of how things are changing.
I would just add to this that something interesting, perhaps in future iterations, to think about is also adding these historic changes as a factor of vulnerability. So currently, we're looking at vulnerability based on their contemporary genetic makeup.
Yes, it's a
but another
snapshot.
factor that can be added to it is also, okay, have this population actually been declining over the past century? So what's their population trajectory?
And use
Yeah,
this to
so I
inform
actually
the predictions. Yeah.
I actually added a slide while Orly was talking about that because it's super interesting to look at the demographic history of populations, which might also be an indicator of future kind of change. So it's really cool.
So in your analysis you mentioned about 100 species, right? That you are applying this to. So maybe this is a bit related with Teresa's question before. Do you see so common patterns of prediction that allows you sort of to predict the prediction that you will get according to I don't know, the biology of the species you have, predictions of mobility and migration rates and things like that?
Yeah,
Do you see these patterns across species? These meta-analyses are really cool.
I mean, I'm in the middle of this analysis. I see patterns. I'm not sure if the patterns make sense yet, so I'm trying to make sure of these patterns. One thing that I'm doing with these data sets is that we've got a really rich body of particularly amphibian data in Europe and Africa, and the data sets are very comparable, so they're sort of all ddRAD-seq kind of data. So I'm primarily focusing on those at the moment, and where we can get estimates of specific traits that are related to the species as well. So we've got kind of estimates of fecundity and dispersal kind of range sizes as well. So that's what I'm going to try and link to the actual outputs with kind of structural equation models to look at kind of causality between vulnerability and these ecological characteristics. And I think with Orly's bats, I mean, Orly's got a wealth of bat data as well. I think it would be really cool to look at some of these bat species together as well. Yeah.
Excellent. So I don't know if there are more questions in YouTube. Check. Yes, there's one here. So from Elika Faust, to what degree can you remove geographic spaces which are not accessible to a species? For example, freshwater fish are very limited in their movements and connectivity.
I mean, yeah, you can just mask out from the environmental data that you put in, you can just mask out regions, I guess, if they're truly inaccessible. You'd have to be careful that you're not masking out potentially accessible areas, I guess. What do you think, Orly? I mean,
Yeah, I mean, I would say from a modeling perspective, best practices are to mask out areas where the species definitely cannot occur. So if you work with terrestrial organisms, it's a bit easier. It's pretty much across their land. You can keep that as an area that they can potentially occur, but that becomes a lot more of an issue when you think about freshwater organisms, or especially marine organisms that are associated with specific strata or levels or depth of the ocean. And if you include suddenly areas that there is no chance they're going to occur, and it ends up with the models over predicting. So it's best practice to actually restrict the area to areas where the species can potentially occur. Now, can potentially occur doesn't mean it's where the species is currently found, because the species may not be found in an area simply because it's dispersal limitation, competition with other species. So many biotic interactions may be missing there. So we don't want to miss where the species can potentially be, but we don't want to include areas where they definitely cannot be.
Yeah. And so I have perhaps one final question. So all these analyses are correlative, right? They find correlations between genetic variation and, of course, and environmental variance. But each there is some complexity in the system, right? Because you have traits that may be more impactful for the survival of species, others that are not. Do you see incorporating knowledge about that? For example, I don't know, I'm working, for example, in seasonal coloration, seasonal coat color change, and this is a major effect, phenotype that is affected by climate change, for example. Do you see this is sort of anonymous, your approach? So you don't need, which is very cool, because you don't need virtually any prior knowledge about that. But do you think that incorporating this, so these eventually massive effect phenotypes that can help to improve predictions, or do you see that as sort of the researcher biasing too much the analysis, and you would prefer to let the data speak for themselves, for itself?
From my perspective, I would say that will be the ideal situation. If we had this kind of data and knowledge, it would be ideal to have the prior knowledge. The reality is that the scope of what we need to understand in terms of vulnerability to environmental changes in a number of species, there is no chance that we'll have this fine scale data for all of them. Yeah. So we do need to have the generalist tools that can give us these more generalized kind of correlative predictions, but they should really be supplemented by more in-depth studies, perhaps of those species that are more interesting in the sense that they're either highly sensitive, or perhaps they're not, and it's interesting to understand why they're not. So it's not that the approaches are contradictory to one each other. I think they're very complementary, and the ideal situation will be to combine everything together.
Yeah, I agree. I mean, I think if you've got specific knowledge about a system, I mean, why not use it in that system? And that was also a bit the motivation for trying to bring in functional genomics in the future as well. So if you've got ideas of sort of deleterious variants, or sort of like beneficial thermoregulatory genes or something like that, would be great. That would be really useful. So yeah, there's lots of scope, lots to do.
Yes. Okay, I don't think there's more questions. So I think we can wrap this up. So thank you very much, Orly and Chris, for this wonderful talk and discussion. I'm very much looking forward to actually using your tool in the future. And yeah, and thank you very much. It's been a pleasure.
All right.
Thank you very much for inviting us.
Yeah, thanks again. Take care.
Cheers. Bye.
Bye-bye.